All right, thank you, Laura. So today is a little bit different from the past 2 days in that today you do have the ability to unmute yourself if you would like to ask a question live, or if you prefer to type your question in the chat, I am happy to read them out to you. Or for Laura, excuse me. So I'll pause just for a moment to see if anybody would like to ask a question live, and if not, I have a few questions for you, Laura, to get things started.
I would also like to point out to everybody that we have Erin's expertise on here as well as a CDWG oversight member and a leader of the VSEPs and GSEPs as well, and also Heidi Ream, who has plenty of expertise as well. So I think between the three of us, we can answer all of your questions.
All right, well, to kick things off, Laura, at what point in the process, so if a group is thinking about forming either a new GSEP or a new VSEP, at what point in the process should they reach out to ClinGen? Like, should they wait until they have their application totally complete, or should they reach out to you earlier? Like, what is the process for that?
I would say that anyone who's interested in establishing either a VSEP or a GSEP should reach out to us immediately. We are here to answer your questions and really make sure that the process is as seamless as possible. So if you are thinking that you might be interested, please reach out to me and the rest of the CDWG Oversight Committee at that email, and we can definitely help you with all of the resources that can make it as pleasant and nice a process as possible. And we can integrate you with the kind of the larger ClinGen group as well, which is kind of one of the reasons that the Oversight Committee is established, to be able to, help groups that are coming in and who want to kind of become part of the process in the consortium.
And we can also help with identifying potential members of the committee, help you sort of figure out who are the experts, how do I go about identifying them, because one of the things that's really critical about these expert panels is that they really represent the global expertise and that we can bring together what may be different types of expertise, but still all surrounding the same disease area. So there might be clinical lab directors, researchers, clinicians who treat patients, and we can potentially help you, identify some of those people if you may only have a subset. Maybe you're a research community, but you might need help identifying clinical lab directors or variant scientists or clinicians to join your group, and we could help with that.
All right, and we have had a question come in through the chat. So as an individual who wants to get involved, this feels quite overwhelming, and I totally feel you on that. That's definitely true. How could an individual kind of get involved in ClinGen at first and choose where they could get and provide the most value?
So as an individual, I think, Amanda, if you are interested in becoming involved with ClinGen, but are maybe interested in seeing how the expert panels operate before you think about launching one yourself, I would recommend becoming a volunteer. You could volunteer in any of several of the different curation activities. And if you go to the ClinGen website, you can, or I think actually on the website that is the web page that's set up for this group, you can click on the Get Involved button, and I think it will take you to a link to the survey that will help you, and actually Erin has put it in the chat. Thank you, Erin. So you can find out more about what the groups do, and you can become a contributing member. You can complete the training in any of these curation activities, and get a firsthand look at what the groups do before you think about any other involvement in the group as well.
And I think if you do have a particular clinical area of interest or expertise, then pick, and there's already a existing expert panel that exists in that disease area. Just simply joining the calls and doing sort of similar to Laura, but then you haven't even committed to doing any specific curation, but you're joining a group that's focused on an area of your interest. You can start to sort of see how they operate and then get involved. I also think if you're wanting to start an area, gene curation is a little easier in the sense that the rules are completely already defined for you. And so you're just implementing ClinGen's existing framework for evaluating gene disease associations, whereas the variant curation expert panels are a little more complex in that you first have to go through this specification of the rules for your gene and disease, and that does take some time to go through that. So if you're not dead set on a specific thing and you just want to kind of get involved in ClinGen, gene curation might be a slightly quicker way to sort of dive into the goal, because the group can immediately start curating using ClinGen's framework.
Yeah. Speaking of too long, another question that came in through the chat, the approval period for VSEPs, they noticed, Laura, you mentioned, is about 18 months. And can you comment on why it takes so much longer for a VSEP to be approved than a GSEP?
Sure. So if you noticed when I was talking through the timeline, the VSEP has the VSEP approval process has these 4 different steps. So the first step is really pretty straightforward and moves pretty quickly, but then it does take some time to work through the ACMG AMP specifications process, though the sequence variant interpretation working group has made a number of recommendations that are applicable to many different groups, particularly with things like allele frequency and other standardized criteria codes. And then as more VSEPs work through the specification process, they each put their specified rules onto both their ClinGen website and on ClinVar, and those are accessible to new groups who are developing. So we are noticing that the process is becoming more streamlined and faster for new groups that are developing. And then the pilot process does take some time as well. So there's time for identifying which of your gold standard variants you're going to use for the classification process, and completing that process, and then also some time to complete the 4th step and just meeting with your groups, establishing a schedule of meetings and review. So we are definitely noticing that it is getting faster, and so I think I wanted to put that up there just to give you an idea of the amount of time that it's taken in the past, but there are definitely some groups who've gone through it much faster than that, but just to give you kind of a realistic approximation of how long it could take.
So it's not that you submit an application and then ClinGen sits on it for 18 months and nobody talks to you for 18 months. It's just that there are multiple back and forth pieces within that process, particularly the pilot process. I mean, one of the groups that I am involved with did 88 variants as part of their pilot, and you can imagine that takes a bit of time. So like all of that kind of goes into it.
All right, so you're actually doing work during the process of the approval. And then the moment you get approved, that first batch, as there instead of 88 variants, immediately gets submitted to ClinVar, and there's like, a major bolus of work that you've done.
Yeah. Now, Heidi, this one you might be able to comment on. So I know that you've been a part of groups that have worked with members across multiple different time zones. So can you share with the group, like, how did you make that work?
Yeah, the most recent group we're working with is the kidney groups. And we have a number of people from Australia, as well as the US and various places. And so they actually split their meetings so that they have some first thing in the morning, like there's one at 8 a.m. East Coast time in the US, and there's one at 5 p.m. East Coast time, but then that translates to more, the late one is morning in Australia, the early one is late at night in Australia. But, then sometimes, different people are more or less able to attend depending on what time of day it is. I think if you were not dead set on a particular disease area because you have expertise or interest in an area and you just want to join as a volunteer, then we can share with you when the actual groups meet, and you could select a group to join based on your time zone. So find one that normally meets in the early morning in the US, which then, for African individuals will be, in the afternoon, things like that.
And we have discussed previously, like if we got a number of people interested, even in an existing group, all from the same or similar time zones, like, could we explore the idea of having different teams working within that group or things like that? So I think we're open to all kinds of possibilities, and we don't want to let your time zone stand in the way of you participating in a group if you are really interested in participating in a group.
Yeah, and groups have changed their meeting times as their membership has evolved. In our Herring Less group, one of our chairs ended up moving to Saudi Arabia. And so we ended up changing the times to accommodate, that individual. So, it sort of depends on who's in the group and trying to make it work for everyone.
Now, a question back to both of you. I'd like to hear both of your thoughts on this. Like, what do you see? Why do we create VSEPs, for example? Like, what is the benefit to the community for going through this process?
Mm-hmm. Do you want me to start, Laura?
Sure.
So, when groups are submitting to ClinVar, if you listened to my talk yesterday, we differ in our interpretation of variants. And even when you get the labs to work together, share their evidence, sometimes we don't always agree with each other. And I made the comment that, is that functional assay really a good assay for that particular gene and disease area? And we really need the experts in those specific areas to be able to say, yes, that's a good assay. It does, in fact, reflect the biological disease, and it's been well validated. And we need experts, particularly researchers in some of those cases, but also clinicians who understand the pathology associated with the phenotype, to be able to, mark those assays valid or not, and how they get scored in the framework. So by developing these disease-specific specifications of the ACMG guidelines, and also, for example, as Stephen was pointing out, what allele frequency should you use to rule out a variant? Well, it depends on your disease penetrance and prevalence. Those are things that experts in that clinical field understand better than just general, group of us trying to curate every variant. So these things are really helpful for experts to come together and agree on, and it really creates better accuracy of variant interpretation. Does that answer your question, Erin?
Yeah, I think that's certainly one answer. Laura, do you have anything else that you would add?
I would also point out that developing these VSEPs enables us to standardize our processes across ClinGen so that they're reproducible. And it also enables us to kind of learn from each other and build more of a knowledge base going forward.
Yeah, and actually it brings up another point, Laura's comment. we can only submit so much information into databases like ClinVar because of patient confidentiality. And so what we find is when we bring the actual groups together that represent the clinical labs and the research programs that are actually testing patients, and they have clinical phenotypes, they're often willing to share that in a limited circle of experts to help justify and document the, the evidence for pathogenicity, when they really don't have enough consent to submit it into a public database like ClinVar. And so there is this process of sharing some of the underlying evidence in a more controlled environment, kind of like clinicians share with each other when they're trying to diagnose a patient. They may share information in the interest of diagnosing a patient. So, a little additional information can be shared amongst that more closed community that really helps with classifying variants. So that's another reason to bring the experts together in this sort of VSEP environment, because we can often share more freely.
Yeah, and I would even add to that, I think another benefit, Heidi, you even mentioned this in your talk yesterday, the VSEPs cannot classify every single variant that's ever been reported or not reported or what have you. I mean, there is a limit to what we as humans can do, but the actual act of specifying the guidelines for that gene and or disease area and kind of putting that out there to the public, that in and of itself is a great benefit, because now anybody in the world can take those recommendations and use them in their own practice. And so, Amanda, back to your point of, as an individual, this kind of all feels overwhelming. Like, how can I, as one person, take any piece or part of this? And at the very least, one thing that you could consider doing is looking at these guidelines as they are available, these disease-specific or gene-specific recommendations, and choosing to use them in your clinical practice, whether it's in a clinical laboratory or even if you're a clinician, looking at reports that you've received or reanalyzing something that one of your patients got years ago. You could use these guidelines and kind of move everyone towards consensus using all of the same rules to classify variants.
That's right.
I would also just like to mention, for people who were not able to hear the talk during the actual talk time, but want to listen to it later, please feel free to reach out to us if you have questions after you've had a chance to listen to it. Yep. We are definitely here to help find a way to involve interested folks in ClinGen.
Yep. And although today's session was focused on starting a new GSEP or VSEP, the reality is, if it seems overwhelming, like, most people don't start their own, they join first and participate, and that's much easier. There's no commitment other than, willing to join calls and offer expertise. Or if you're being trained as a volunteer, then we do ask you to try to commit to some level of commitment for some variants, but it could be like one a month, one variant a month, or one gene every few months or something. They're not, and we understand if you got in over your head and that's not possible. There's nothing bad will happen if you can't deliver. We really want to encourage participation in any way that you feel you can manage.
Right. And so I think, how to look at the already established expert panels and see if any of those seem like an area of interest to you. And if that's something that you're interested in becoming involved in, we can certainly facilitate that for you.
Yeah.
All right. Well, thank you guys. You can have a quick 2-minute break and we'll see you back in the main room.